There is an organ inside you right now performing over 500 distinct biological functions simultaneously. It filters, it builds, it destroys, it stores, it detects, it signals. No machine ever built by human hands comes close to replicating what it does. And yet, when people think about the organs keeping them alive, they almost never think of it first. The heart gets the poetry. The brain gets the philosophy. The liver gets almost nothing until it fails. That omission is not just a cultural curiosity. It reflects a deep misunderstanding of how the human body actually works. Because if you had to choose one organ whose loss would trigger the fastest and most catastrophic collapse of nearly every other biological system, the liver would be the strongest candidate. Not the heart, not the lungs. The liver. What follows is not a warning about alcohol. It is something more fundamental, the story of a biological architecture so sophisticated, so deeply embedded in the chemistry of life, that understanding it changes how you see the body entirely. The liver sits in the upper right quadrant of the abdomen, just beneath the diaphragm. Thus, in a healthy adult, it weighs between 3 and 3.5 pounds, making it the largest solid organ in the body. It is reddish-brown, dense, and divided into 2 primary lobes, the larger right lobe and the smaller left lobe, along with 2 smaller lobes at its back surface. From the outside, it looks almost unremarkable. A dense, quiet mass of tissue tucked under the rib cage, doing nothing visible, making no sound, offering no pulse you can feel with your hand. But the moment you look inside, the picture changes completely. The liver has something almost no other organ in the body possesses: a dual blood supply. Most organs receive blood from one source, an artery carrying oxygenated blood. The liver receives blood from 2. The hepatic artery delivers oxygenated blood in the conventional way, but the portal vein delivers something far more interesting: blood that has already passed through the digestive tract, carrying with it every nutrient, every toxin, every drug, every byproduct of digestion that was absorbed through the walls of the intestine. This means that everything you eat and drink passes through the liver before it reaches the rest of your body. Everything. The liver is not downstream of digestion, it is the checkpoint at its exit. And that single architectural feature explains an enormous portion of what the liver does and why it matters so profoundly. To understand how the liver processes this enormous chemical load, you have to go inside its tissue, past the surface, past the lobes, down to the functional unit that actually does the work. That unit is called the hepatic lobule. The liver is composed of somewhere between 50,000 and 100,000 of these lobules, each one a hexagonal column of tissue roughly 1/25 of an inch in diameter. At the center of each lobule runs a central vein. Around each lobule's 6 corners run portal tracts, small bundles carrying a branch of the portal vein, a branch of the hepatic artery, and a bile duct. Blood from both the portal vein and hepatic artery enters the lobule at its edges and flows inward through tiny channels called sinusoids, eventually draining into the central vein, which then carries it toward the inferior vena cava and the heart. Along those sinusoids live the liver's primary working cells, hepatocytes. These cells make up roughly 80% of the liver's total mass. They are metabolically extraordinary. Some of the most chemically active cells in the entire human body. Each hepatocyte is in direct contact with the blood flowing through the sinusoids. There is no barrier, no membrane wall holding blood back. The blood washes past the hepatocytes almost directly, and in that contact, the chemistry of life happens in ways that no other tissue replicates. Here is where the real depth of what the liver does begins to become clear, and where most textbook descriptions fall short. The liver's functions are usually listed. That list sounds impressive in a mechanical way: metabolism, detoxification, protein synthesis, bile production, storage, immune surveillance. But a list does not capture the interdependency. It does not capture what it means for a single organ to be the hub through which almost every metabolic pathway in the body runs. Take carbohydrate metabolism. When you eat, glucose floods through the portal vein into the liver. Hepatocytes absorb it and convert it into glycogen, a storage form of glucose, holding it in reserve. When blood sugar drops hours later, those same hepatocytes break the glycogen back down, releasing glucose back into circulation. The liver is not just responding to the body's energy needs, it is actively managing them, smoothing out the spikes and valleys of intake and demand in a way that keeps the brain and every other glucose-dependent organ supplied continuously. But when glycogen stores are full, the liver does something else. It converts excess glucose into fatty acids, which are packaged into lipoproteins and sent out to fat tissue for long-term storage. This process, called de novo lipogenesis, is one of the reasons that dietary patterns have such complex effects on body composition. The liver is not a passive conduit. It is an active metabolic decision maker, constantly interpreting chemical signals and choosing between pathways based on the body's current state. The same layered logic applies to fat itself. The liver synthesizes cholesterol, including the forms packaged into LDL and HDL particles, and regulates its distribution across the body. It processes incoming dietary fats, manufactures bile acids from cholesterol to facilitate fat digestion in the intestine, and recycles those bile acids through a circuit called enterohepatic circulation. Cholesterol, it turns out, is not simply consumed from food and deposited in arteries. Most of it is synthesized, regulated, and distributed by the liver. This is why drugs designed to lower cholesterol, statins for example, target a liver enzyme called HMG-CoA reductase, not any process in the cardiovascular system directly. Now consider the liver's role in protein. The majority of the proteins circulating in human blood are made in the liver. Albumin, the most abundant protein in blood plasma, is produced exclusively by hepatocytes. It carries hormones, fatty acids, and drugs through the bloodstream, and it maintains osmotic pressure, the force that keeps fluid from leaking out of blood vessels into surrounding tissue. When the liver fails and albumin production drops, fluid accumulates in the abdomen and legs, a condition called edema, visible evidence of a protein that was always there, always essential, and rarely noticed until it disappears. The liver also produces most of the clotting factors, the proteins the body deploys to stop bleeding. When liver function deteriorates severely, clotting capacity falls with it. Patients with advanced liver disease can bleed from minor injuries in ways that are difficult to control, because the molecular machinery that would normally seal the damage was never assembled. This extends to something subtler but equally important. The liver synthesizes acute phase proteins, molecules that rise rapidly in the bloodstream during infection or injury, and help coordinate the immune response. The liver is not just a metabolic organ. It is an active immunological participant, producing molecules that signal danger, recruit immune cells, and modulate inflammation throughout the body. There is a category of liver function that is perhaps the most intuitive, but also the most often misunderstood: detoxification. The liver does detoxify, but the word is used so loosely in popular culture, attached to products, diets, and cleanses, that its actual biological meaning has become obscured. In the liver, detoxification refers to a specific set of enzymatic reactions that chemically transform substances into forms the body can excrete. These reactions are divided into two phases. In phase 1, a family of enzymes called cytochrome P450 enzymes modifies the structure of molecules, introducing or exposing chemical groups that make the substance more reactive. In phase 2, those modified substances are conjugated, chemically linked to other molecules that make them water-soluble and therefore excretable through bile or urine. This process handles alcohol, which is converted first to acetaldehyde, a compound more toxic than ethanol itself, and then to acetate, which the body can safely process. It handles most pharmaceutical drugs, which is why drug dosages must account for how quickly the liver metabolizes them. It handles ammonia, a toxic byproduct of protein metabolism, converting it to urea for excretion by the kidneys. And it handles a wide range of compounds that arrive through food, water, and the environment. What is worth understanding clearly is that this system has limits. The cytochrome P450 enzymes can be saturated. They can be inhibited by certain compounds that compete for the same active sites, which is one mechanism behind drug interactions. And they can be induced, accelerated, by repeated exposure to certain substances, which is why people who regularly use certain medications or consume alcohol regularly process them differently over time. The liver, in other words, is not a static filter. It is a dynamic, adaptive, and sometimes overwhelmed system that changes its own chemistry in response to what it encounters. Alongside all of this, the liver is the body's most significant storage depot. It holds glycogen as discussed, but also fat-soluble vitamins, A, D, E, and K, and vitamin B12, storing enough in some cases to last years. It holds iron in a form bound to a protein called ferritin, releasing it in response to the body's needs. The liver holds copper. It holds reserve concentrations of metabolic intermediates that other tissues cannot store efficiently. This storage function has profound implications for nutrition and disease. Vitamin A toxicity, for instance, develops when the liver's storage capacity for retinol is exceeded, not from food in typical circumstances, but from high-dose supplementation. Iron overload in the liver, in conditions like hemochromatosis, can lead to fibrosis and eventual cirrhosis even in people who do not drink. The liver's role as a reservoir means that what the body accumulates quietly over years can eventually produce consequences that appear suddenly. The liver also has a property that makes it nearly unique among the solid organs of the human body. It can regenerate. Remove up to 70% of a healthy liver, and the remaining tissue will grow back to approximately its original size within weeks. This is not an approximation. It has been demonstrated surgically in the context of living donor liver transplantation, where a portion of a healthy donor's liver is removed and transplanted into a recipient. And both the donor remnant and the transplanted portion grow to functional size in the weeks that follow. The mechanism involves a precise interplay of growth factors, cytokines, and cellular signaling pathways. Hepatocytes, which under normal conditions divide only rarely, re-enter the cell cycle in response to the loss of liver mass. Research continues to explore the precise signals that initiate this response and, crucially, the signals that terminate it once the appropriate size is reached. What is clear is that the liver possesses a regenerative capacity that goes far beyond simple wound healing. It is a capacity for the restoration of entire functional architecture. This property has ancient recognition. Greek mythology gave us Prometheus, chained to a rock while an eagle ate his liver each day, only for it to regrow overnight. The myth predates any understanding of hepatocyte biology by 2 millennia. Whether the Greeks observed something in animals or arrived at the image through other means, evidence suggests they were describing something real, even if they had no framework to explain it. There is still no full consensus on many aspects of how the liver manages its own regenerative limits, or precisely how it coordinates metabolism across so many parallel pathways without conflict. Scientists are still investigating how hepatocytes communicate with neighboring non-parenchymal cells, the Kupffer cells, which are resident immune macrophages embedded in the liver's sinusoidal tissue, and the stellate cells, which regulate fibrosis and in some contexts drive the scarring that leads to cirrhosis. Kupffer cells are an important and underappreciated part of the liver's biology. They constitute the largest fixed population of macrophages in the body, positioned directly in the path of portal blood flow. every bacterium that slips past the gut barrier, every endotoxin released by intestinal microorganisms passes through the liver's sinusoids and encounters these cells. Kupffer cells filter, engulf, and respond, activating inflammatory cascades when necessary, tolerating harmless material when they can. The liver is, in this sense, a surveillance organ positioned at the interface between the outside world, everything that enters through the gut, and the systemic circulation. Studies indicate that the relationship between the gut microbiome and liver health is closer than previously understood. Changes in the composition of intestinal bacteria can alter the chemical environment in the portal vein, affecting how much inflammatory signal reaches the liver and how Kupffer cells respond. This bidirectional relationship, gut shaping liver, liver responding to gut, is an active area of research with significant implications for understanding conditions like non-alcoholic fatty liver disease, which has become increasingly prevalent and which research continues to connect to both metabolic and microbial factors. The liver is a biological system of almost impossible breadth. It synthesizes, it metabolizes, it stores, it secretes, it filters, it regenerates, it signals. It operates continuously, without pause, at a scale of chemical complexity that makes the most advanced industrial chemical plant look simple. And it does all of this silently, with no sensation, no perceptible rhythm, no signal to the conscious mind that anything is happening at all. That silence is worth sitting with for a moment. The liver does not ask for attention. It does not announce its efforts. A damaged liver can lose 60 or 70% of its function before any symptom appears, because the organ's reserve capacity is enormous and its backup systems run deep. By the time the skin turns yellow with jaundice, by the time fluid fills the abdomen, by the time fatigue becomes unmanageable, significant, often irreversible change has already occurred. The liver is not just underrated in a cultural sense. It is biologically designed to be invisible, to absorb damage quietly, to compensate without complaint. There is something profound in that design. The organs we notice are the ones that make themselves felt. The heart that races, the lungs that burn, the muscles that ache. The liver, by contrast, asks for nothing. It processes everything you give it. The food, the alcohol, the medications, the environmental exposures, and transforms them, neutralizes them, packages them for distribution or elimination, and continues without a signal back. Its silence is not indifference, it is extraordinary competence. And in that silence lives a kind of architectural truth about the human body. Not every system announces its importance. Not every critical structure is loud or visible or emotionally resonant. Some of the most essential functions in the body are the ones running deepest in the background, the ones whose presence you only register by imagining their absence. The liver is the body's answer to complexity, a single organ that learned over hundreds of millions of years of evolution to be the center of everything chemical in the body without ever demanding to be noticed. The architecture is not incidental. It is, in its own quiet way, the most sophisticated thing inside you. And perhaps that is the most fitting place to leave this. Not with a warning, not with a statistic, but with a quiet recognition that inside you, right now, an organ the size of a small dictionary is running the chemistry of your entire existence, silently, without complaint, without credit. The liver does not need your attention to do its work, but it deserves it. If this made you see the human body differently, if it changed even slightly how you think about what is happening beneath the surface, then it did exactly what it was meant to do. Leave a like. It takes a second, and it helps this kind of science reach the people who need it most. And if you want to keep going deeper into the hidden architecture of the human body, subscribe to Anatomy Unlocked. Every video is built the same way. No shortcuts, no hype, just the real science of what you are made of. The body has more stories like this one. We are just getting started.